to dear aspirants of the NET. Today we are going to discuss basics of immunology. Immunology, as you know, is the science that deals with the defense against the invading pathogens, most notably microorganisms, viruses and other infectious agents. Our immune system is a feature of vertebrates, and this particular resistance to protect the individual from infectious agents or the diseases is known as immunity. Immunity has both specific as well as non-specific components. The less specific or non-specific components, which are known as innate immunity, provides the first line of the defense against the infections. It does not have memory response, but this is the first barrier that prevents the entry of pathogen into the body of an individual. The immunity, which is the resistance of the individual against the invading pathogens, is because of a very intricate and well-developed immune system. This immune system is a body-wide network of cells and organs which has evolved during the evolution to defend the body against the attack by various foreign invaders. This immune system generates a very strong, prompt, appropriate, effective and very self-limiting response that successfully eliminates the pathogens from the body. This particular system depends upon a very coordinated and dynamic communication and interactions among the millions of cells which comprises the immune system. Along with it, it is comprised of lymphatic system, primary, secondary and tertiary lymphoid organs. There are variety of cells which plays an important role in the various features and the capacities of the immune system, which includes B and T lymphocytes, macrophages, NK cells, monocytes, neutrophils and dendritic cells. All these cells provides us a very strong defense against variety of foreign molecules and microorganisms. We will be discussing the role of all of these cells one by one in the coming slides. Now, whenever a foreign organism enters into our body, be it a pathogen, be it a virus, the first process that our immune system does is to recognize. And the first step in the recognition is whether it is self or non-self. Our immune system makes a deliberative effort to recognize whether it is self or non-self. Thereafter, it recognizes specific patterns on the surface of a particular foreign microorganism. Thereafter, it generates a response that will eventually eliminate or neutralize that foreign substance or microorganism. Now, it might have a particular memory response. That means it recognizes the pattern and the kind of organism that has entered once. Thereafter, the subsequent exposure of the same microorganism would be generating a very strong, faster and more intense response, and the same antigen will be eliminated in a lesser time in a much rapid pace. So typically, an immune response is having two particular stages. The first is recognition and second is response. And thereafter, a particular immune response would have a memory, so that whenever second time the same antigen enters into our body, it will be eliminated much faster. Now, if we see actually what is an antigen, there are variety of substances that may serve as antigen. For example, in this particular question, it is being asked an antigen is a protein molecule that helps the defend the body against the diseases. It's a kind of white blood cell. It's a specific molecule present on cell, or it is a foreign molecule that evokes a specific response by a lymphocyte. This fourth option is right. A foreign molecule that evokes a specific response by a lymphocyte. Here one thing is very important. Sometimes the same antigen is more appropriately is being called as immunogen. Immunogen is that particular foreign substance or molecule that evokes the immune response. Now, if we see there is an immunogen that can evoke the immune response, there are certain features which decides the immunogenicity of a particular antigen. The immunogenicity of a particular antigen depends on variety of entities. It might be the foreignness, the molecular weight, its chemical structure, chemical complexity of the molecule. So, in this particular question, if you see which of the following does affect immunogenicity of a substance, there are certain options like making a molecule more foreign, making a molecule larger, increasing chemical complexity by adding amino acids, and D, all of the above. Here the right option is D, all of the above, because as we increases the foreignness or as we increases the phylogenetic distance between the recipient and the antigen, the extent of immunogenicity will increase. Likewise, larger the molecule in the size, most notably those particular pathogens whose size is more than 1 lakh Dalton, they are a strong immunogen. Similarly, if you compare two particular pathogens, one particular pathogen or immunogen which is having more structural heterogeneity in terms of amino acids, not only in the type of amino acids, but the aromatic amino acids, if the extent of aromatic amino acid increases, the immunogenicity will also increase. Along with it, the chances of a particular pathogen to get processed faster would also make it a strong immunogen. So there are certain factors which increases the immunogenicity of the substance. This is being asked in the particular question. Sometimes they do ask stuffs like, as you see in this particular question, the antibiotic penicillin is a small molecule that does not induce antibody formation. Here it means to say that it is not generating any immune response. However, penicillin binds to serum protein and forms a complex that in some people induces antibody formation, resulting in an allergic reaction. Penicillin is therefore, it is an antigen, option B haptane, it's an immunogen, or it's both antigen and haptane. Here the concept that is being asked is there is an entity which by itself cannot generate immune response, but when it gets bind to something else, it is generating a stronger immune response. How it becomes possible and what the kind of molecule it is. Such kind of molecules are known as haptanes. By themselves they cannot evoke the immune response, but if they are clubbed with or if they are conjugated with certain carrier molecules who are bigger molecules, they have features of immunogenicity as we have discussed in the earlier section. If they are clubbed with or conjugated with those carrier, they can evoke immune response. Such molecules are called as haptanes. They do not possess immunogenicity, but they do possess antigenicity. Haptanes are important because so many drugs, so many molecules, so many enzymes, they belong to this particular category. Similarly, if we see, there is a specific question on haptanes. Haptanes, they require carrier molecules to be immunogenic, react with specific antibodies when homologous carriers are employed, interact with a specific antibody even if the haptane is monovalent. So here, the first particular part we have already discussed, that they would require carrier molecules to be immunogenic. Otherwise, by themselves, they cannot evoke the immune response. Now, when a particular antigen enters into the body of an individual, actually antigen is a very larger molecule. It's a macromolecule. So the entire molecule or entire immune response is not targeted against entire organism. There are specific signature sequences in terms of amino acids. They are like certain patterns which are being recognized by the immune system. Now those specific patterns which are present on the surface of antigen, they are called as epitopes. So epitopes are specific regions against which immune response is generated. This is being reflected in this particular question that you can see. Epitope is the discrete region present on the antigen that interacts with option A, FAB, that means fragment antigen binding. Option B, antibodies heavy and light chain. Option C, T cell receptor. Option D, all of the above. Actually, when immune response is generated, specifically by B cells, they will generate antibodies. And these antibodies will have a particular region that will specifically bind with the epitopes. Technically, that is being called as paratope. So, there is a specific region present on the antigen which is recognized by our immune system to generate immune response. Now, those immune molecules which are being produced against the pathogen, they will also have a specific region that will particularly interact with the epitope. Such regions would be called as paratopes. And these paratopes are present on the heavy and light chain of the antibody molecule. This we will see in the diagrams in the coming sections. Now, the entire immune system comprises of variety of cells, and all these cells works in tandem with each other. They work in coordination with each other to bring about the required effect. Now, all the cells of the immune systems, they are derived within the bone marrow by the process of hematopoiesis. And all these cells of the immune cells, they are being derived by a phenomena that is known as hematopoiesis, and these cells are called as hematopoietic stem cells. These hematopoietic stem cells give rise to two lineages. One is lymphoid lineage and another is myeloid lineage. The lymphoid lineage will give rise to B lymphocytes, which will produce antibodies. They will give rise to T helper cells that will give rise to cytokines. They can give rise to TC cells or cytotoxic T cells. The job of the cytotoxic T lymphocytes is to specifically kill the altered self cells. Altered self cells are those cells which are our own cells, but they have now altered because of either infection by a virus or might be they have become cancerous. In both these situations, this particular cell is altered and now present a threat to the individual. Killing the altered self cell is the job of the CTLs. And likewise, there is another cell that is known as NK cell or natural killer cell. This natural killer cell also kills altered self cells, albeit non-specifically. They can also give rise to the lymphoid progeny. They can also give rise to dendritic cells. Dendritic cells are having long dendrites or cytoplasmic processes, and because of which they have gained this particular name, dendritic cells. These are phagocytotic in nature. They can phagocytose or engulf the antigen, and then a part of that antigen would be presented to T helper cells. Such cells which can engulf the antigen and a specific signature sequence that we called epitope would be presented to T helper cell. This particular phenomena is called as antigen presentation. And all those cells which are capable of doing this would be called as antigen presenting cells. Now we are coming to myeloid progeny. The myeloid progeny, which is being derived from hematopoietic stem cells, they can again give rise to monocytes. Monocytes will circulate in the body for a few hours and then they will settle down in different organs. And there they will be now called as macrophages. Macro means big and phages means they are also capable of phagocytosis. So there are variety of macrophages spread throughout the body. They can also give rise to, the myeloid progeny can also give rise to granulocytes. There are three different kinds of granulocytes: eosinophils, basophils and neutrophils. They have attained these names because of their nature. They are being stained by acidic dyes, then they are called as acidophils or eosinophils. If they are stained with basic dyes, they are being called as basophils, and if they can be stained with both, they will be called as neutrophils. The neutrophils again are phagocytic in nature. Eosinophils, they provide us immunity against parasitic infections. Basophils, they generate allergic manifestations. They have certain granules which are rich in histamines and other substances. Now, the function of various kinds of B cells we have already discussed earlier. B cell, they will produce antibodies, And these antibodies will specifically eliminate the antigen by a variety of mechanisms. T helper cell will produce cytokines. Here, it is worth mentioning that cytokines are very important molecules. Cytokines regulates the intensity and duration of the immune response. And these cytokines are secreted by T helper cells. There comes TC cells. We have discussed these cytotoxic T cells. They will kill the altered cells. Specifically, a particular kind of TC cell will kill a particular kind of altered cell only. NK cell will also do the same job, but they will do it non-specifically. That means if a NK cell will come across any kind of altered cell, it will kill it. Whereas a TC cell will kill only a specific kind of altered cells for which it is highly specific. Macrophages and dendritic cells, along with B cells, along with neutrophils, they all are phagocytic in nature. They will phagocytose the antigen, and a specific signature sequence of that antigen, that is known as epitope. Epitope would be presented to T helper cells, and these cells would be categorically called as antigen presenting cells. Now there is a model question on this particular thing. Tissue macrophages begin their lives as neutrophils? No. Have short lifespans because they self-destruct after engulfing the foreign invaders? It is not like that. Originate from monocytes that leave the circulation and enter the tissue. This is correct. And are most effective against parasites? Actually, it is not. Mostly, it is eosinophils that are most effective against parasites. So here, the fact that is being asked is, macrophages are derived from which kind of cells? Macrophages are derived from monocytes. Monocytes are derived from myeloid progenitor cells. These will circulate for some time in the body and then they will eventually differentiate into macrophages. This particular fact is being asked in this particular question. We have discussed antigen presenting cells. So here it's a very simple question that is being asked. Antigen presenting cell in higher organisms is T cells, macrophages, megakaryocytes, eosinophils. Actually, antigen presenting cell in higher organisms, among these four options, it is macrophages. But we should remember there are other cells also which are antigen presenting cells. For example, dendritic cells are there. They are also APC or antigen presenting cells. B cell can also do. B cell can produce antibodies and they can also serve as APCs. And similarly, dendritic cells are also antigen presenting cells present in higher organisms. Sometimes they may give two options, macrophages and dendrites, and then they will give both macrophages and dendrites. Such kind of questions can also be there in the exams. Now if we see in this particular question, the examiner wants to ask bigger thing. He wants to know whether you know the function of all the cell types of the immune cells or not. Here you have to identify the mismatch between the cell type and the description, which is the function of that particular cell, whether it is the right function or it is not. Eosinophil, body defense against parasites, actually it is right. Kupffer cells, they are the macrophages found in Lear, right? Dendritic cell, an antigen presenting cell that is not phagocytic. And fourth, mast cells, these are the cells which are first to arrive at the site of inflammation. This is also right. So the question here is, the mismatch here is dendritic cells. Any antigen presenting cell has to be phagocytic, and here it is written an antigen presenting cell that is not phagocytic. So this is not a right option. So we have to remember any antigen presenting cell has to be phagocytic. Now, if we look into the types of the immunity which is the resistance of an individual against foreign invaders, it can be of two kinds. We have already discussed that it might be highly specific, and in that case it will be known as acquired or adaptive immunity. But the first immunity with which we are all born with, that is known as innate immunity. The innate immunity is the first line of defense that prevents the entry of pathogen into our body. It is non-specific, it does not have memory response. But on the other hand, the acquired immunity will develop only when antigen comes into our body or there is an antigenic challenge to a particular organism. One of the remarkable feature of acquired immunity is that it is having immense diversity. That is, immune response can be generated against vast array of molecules. For example, we can generate immune response against trillions different kinds of molecules or foreign microorganisms. This high level of diversity we have. Along with it, it is highly specific. For one kind of molecule or one kind of organism, we will produce one kind of molecules. For different kind, we will produce different kind of molecules. So we have a very high specificity by means of acquired immune response. And most remarkable feature is memory. It recognizes that one particular kind of organism has entered into our body. We have wiped it out. Might be 20 years back, might be 50 years back. And this immunological memory is being exploited in the process of vaccination. In the case of vaccination, we'll give a very small dose of either live or attenuated antigen. Thereby, an individual generates an immune response. That would be primary immune response. It will develop memory cells, or we will develop immunological memory. And when the same antigen will come into our body naturally anytime during our lifespan, we will generate a very strong immune response against that, and we will wipe out that particular immunogen in a much faster and rapid manner. This is the fact, this is the immunological memory that we exploit, that we use in the case of vaccination. So it is very important thing. Now there is a question, which of the following could be considered a non-specific defense? Option A, intact skin that creates a barrier that cannot normally be penetrated by bacteria or viruses. Option 2 is secretion from sebaceous and sweat glands. Give the skin an acidic pH that prevents bacterial colonization. Option 3 is, or option C is, tears, saliva and mucus secretions that contains lysozymes. And we know that lysozymes are the enzymes that digest the bacterial cell wall. Here, the right option is D, all of the above. So, the skin, our sweat glands or the sweat, our tears, saliva, mucus secretions, all these are the components of the non-specific or innate immunity. Now, the components of the innate immunity includes 4 main barriers. The first one is anatomic barriers like skin. Outside we have a skin cover over us, and that does not allow entry of any pathogen into our body. Internally again we are lined. Right from the mouth we are lined internally. All of our respiratory tracts, our digestive tract, our genitourinary tract, all these are lined internally by mucous membrane. So even if a pathogen enters into our body either by air or water or food, actually it just remains confined within the pipes. And these pipes, be it a respiratory tract or a gastrointestinal tract, they are internally lined by mucous membrane. And these mucous membrane secretes mucus in which antigen is trapped, and by the movement of cilia, it is being eliminated out of the body along with the excreta. So even if pathogen enters into our body via food or air or water, actually it is being propelled out of the body because of internal mucous membrane linings. Then we have physiological barriers that includes pH, temperature, complement system. So what happens? Our body temperature is 37 degrees centigrade. So all those pathogens that survives at 27 or that survives at much higher temperature above 38 or 39, actually we are not appropriate host for them, and we naturally becomes immune against all those particular kind of organisms. Similarly, the pH, when a pathogen enters into our body, there are changes in the pH during the digestive processes, and that particular pH changes are very extreme, and they are not being tolerated by microorganisms, and they die because of that. Then there comes phagocytosis. Phagocytosis is a phenomena of engulfment of the pathogens by specific kinds of cells. Such cells are called as phagocytes. In our body, most prominent phagocytic cells are macrophages, neutrophils, dendritic cells, and even B cell can also phagocytose the antigen, albeit by a different phenomena that is called as receptor mediated endocytosis. So the engulfment of the pathogen is also being done by specific cells which are known as phagocytes. There comes inflammatory barriers. Inflammation is a localized immune response that is being generated against pathogenic organism or tissue injury. Inflammation are having 4 cardinal signs like redness, swelling, pain, rise in temperature, and sometimes there might be involvement of there might be a transient or temporary loss of function. All these are the cardinal signs of inflammation, and sometimes they do ask questions on such particular issues. There are certain mediators, there are certain serum proteins which can bring about the inflammation, and such molecules are known as inflammatory molecules. For example, certain complement proteins, certain cytokines like interferon gamma, they all are capable of generating inflammation in our body. Inflammation is a positive term and it prevents us from variety of pathogens and tissue injury. Sometimes if we see a comparative account of innate and acquired immunity, innate immunity represents the first line of defense because it comes first. Our skin, our mucous membranes, they are the first entities which come against the pathogens. It is non-specific. All these features are present. This innate immunity is present right from the birth. We are born with this resistance right from our birth, but there is no memory response. And whenever innate immunity is working, actually it will take the same time. Whether the antigen is coming for the first time, second time, or another time, it will take same time to eliminate the antigen because it is non-specific and it is limited. Mostly innate immunity is a feature of lower forms of life, or non-vertebrates or stuff like that, and it is mostly instant. On the contrary, if you see acquired immunity, it is the second line of defense, comes after first line of defense. These two works in conjunction with each other, where first line of defense is not able to prevent the entry of pathogen, second line of defense would come. It is highly specific for a specific kind of pathogen, a specific kind of immune response, and molecules are being generated. It develops after the exposure to the antigen, whereas innate immunity is present right from the birth. Whether antigen comes or not, we are having skin. But immune molecules in the case of acquired immunity, they will be developed only after pathogen enters into our body. They can recognize between self and non-self. It is one of the important feature. So immune response would always be generated against non-self entities, and we will not generate immune response against self molecules. It is highly diverse. As we have already discussed, we can generate immune response against trillions of molecules and microorganisms. Sometimes even if you generate a new molecule, we are already having immune molecules against those particular newly designed molecules. This is the diversity of our immune response. And one of the important feature is memory. If an antigen enters into our body for the one particular time or for the first time, it will take particular time to eliminate that antigen. But when the same antigen comes for the second time, it will be eliminated much faster in lesser time because of the immunological memory. So whenever there is immune response is being generated, it will produce memory cells, and these memory will wipe out the antigen whenever it will come. Suppose after 15 days or even after 50 years, it will be eliminated very fast. It is an important feature of acquired immunity. Now in this particular question it is being asked, phagocytosis is a feature of acquired immunity, innate immunity, both innate and acquired, or humoral. So the right option is, it is a feature of innate immunity, because whenever a particular phagocytic cell engulfs a particular antigen, it does it non-specifically. Any particulate substance can be engulfed by the phagocytic cells, and thereby phagocytosis is non-specific and it is a component of innate immunity. Now, immunity due to generation of antibodies belongs to which particular type of immunity? Innate immunity, acquired immunity, cell-mediated immunity, both innate and acquired immunity. So antibodies are generated specifically against a particular kind of antigen. That means a specific immune response is being generated, and specificity is a hallmark of acquired immunity. So here the right option is B, that is acquired immunity. Now we will be discussing certain lymphoid organs. Lymphoid organs are the organs or the sites or the tissues where the development, maturation and differentiation of the lymphocyte takes place. Overall, lymphoid organs can be categorized into two classes. The first one is primary lymphoid organs, second one is secondary lymphoid organs. Primary lymphoid organs are also called as central lymphoid organs, and secondary lymphoid organs are also called as peripheral lymphoid organs. Now there is a basic distinction between the primary lymphoid organs and secondary lymphoid organs. Let me make it clear. Primary lymphoid organs are the sites or tissues where the development of lymphocyte takes place, whereas secondary lymphoid organs are the sites or tissues where the interaction between the antigen and the lymphocyte takes place. So development, hallmark of primary lymphoid organ. Interaction with the antigen is a hallmark of secondary lymphoid organ. One interesting thing is that within the primary lymphoid organs, lymphocytes are generated. They do generate antigenic specificity. But whatever development is taking place in primary lymphoid organs, it is antigen independent. That is, there is no involvement of antigen. Whereas secondary lymphoid organs, whatever sequence of events are there in the secondary lymphoid organs, they are highly dependent upon the presence of antigen. And that is why this entire phase of development is called as antigen dependent phase of lymphocyte development. All these things takes place in secondary lymphoid organs. Now in this particular question, it is being asked, primary lymphoid organs are, they are efficient in exposing T cell to foreign antigens. They are the primary sites of antibody synthesis and maturation. They will filter blood and trap blood-borne antigen. Option D, provide the microenvironment for the synthesis and maturation of T and B cells. The right option is D, because primary lymphoid organs are the sites that will provide the conducive microenvironment for the development and maturation of T and B cells. Now in this particular question it is being asked, which of the two organs are the example of secondary lymphoid organs? Bone marrow and spleen, spleen and lymph nodes, thymus and bone marrow, spleen and malt. So secondary lymphoid organs are those organs where the interaction between the antigen and the lymphocyte takes place. So in this particular thing, the right answer is D, that is spleen and malt. MALT stands for mucosal associated lymphoid tissues. Now here comes a very interesting thing. High rate of apoptosis is observed in primary lymphoid organs, secondary lymphoid organs, both primary and secondary lymphoid organs, and D, spleen. Actually it is very interesting thing. Why there is apoptosis taking place in any kind of lymphoid organs? Let me first give the right answer. High rate of apoptosis is observed in primary lymphoid organs. The primary lymphoid organs for suppose mammals or human beings, there are two primary lymphoid organs. Bone marrow, that is for the development of B lymphocytes, and thymus, that is for the development of T lymphocytes. Now what happens, interestingly, if you see within the bone marrow or the thymus, more than 90 to 95% of the cells, they die. They do not come out as a mature antigen specific lymphocytes. They undergo apoptosis. There are a variety of reasons for it. If a particular lymphocyte recognizes the self molecule, it will not be allowed to develop further and it will die by apoptosis. If a lymphocyte does not produces a specific receptor that is productive, in that case that cell will also die. only those cells which recognizes non-self molecule, they are having rest of the features in appropriate manner, only those lymphocytes will develop and mature further. So 90 to 95% of the cells which fails to achieve all these features, they will die within the primary lymphoid organs, and that is why very high rate of apoptosis is observed in primary lymphoid organs such as bone marrow and thymus. Lymphocyte generates antigenic specificity. It is a very important thing. Where or at which stage lymphocyte generates the antigenic specificity? Option A, after interaction with antigen. Option B, prior to antigen interaction but after the entry of the antigen into the body. Option C, even before the entry of antigen into the body. Option D, according the type of epitopes which are present on the antigen. Interestingly, we produces lymphocytes which are highly antigenic specific even before the entry of antigen in the body. So here the option C is right, and the important fact which we should remember is that our immune system is that much capable, that much well developed, that we develop lymphocytes which are having antigenic specificity, means they are particular for particular antigenic epitope even before the entry of the antigen, means we are already well prepared against variety of antigens which may enter into our body. So in this case, we have to remember that our immune system is that much well developed that we develop lymphocytes even before the entry of the antigen into our body. Now there comes a phenomena that is called as primary and secondary immune response. When a particular antigen enters into our body for the first time, the immune response that we generate, we produce suppose antibodies. So that is called as primary immune response. Now if the same antigen is deliberately injected into an individual so that the same antigen enters into the body for the second time, the immune response that would be generated now would be called as secondary immune response. Now here, whether the antigens comes for the second time, third time, fourth time or nth time, it will be called as secondary immune response. Because of the immunological memory, what happens is, if primary immune response takes 14 days to eliminate the antigen completely from our body, the secondary immune response will do the same in 8 to 9 days. So secondary immune response is much faster, it is much stronger. The kind of antibodies or the immune molecules that are being generated, they are having high specificity compared to the primary immune response, and this particular thing is being used in the case of vaccination. And that is why if a person is vaccinated against a particular pathogen or particular antigen, whenever natural infection of that particular antigen occurs in the body, our immune system will eliminate that particular antigen in much faster pace, in a much stronger manner. That is why vaccination is being recommended. Now there is a question that is being asked here. During a secondary immune response, means what happens during a secondary immune response? Option A, selected B generate antibody producing effector B cells called plasma cells. The stricken individuals may become ill. About 10 to 17 days are required from the exposure to maximum effector response. And option D, the generation of effector cells begins with memory cells produced during the primary immune response. Yes, the option D is right. During a secondary immune response, the immune response is being generated from cells which are already being developed during the primary immune response, which are being called as memory cells. Now the memory cells will come into action. They will eliminate the antigen in a much faster manner. Here we should be able to distinguish between two terms. One is known as neo-lymphocyte, another is memory cell. When the antigen interacts with the lymphocyte for the first time, this particular lymphocyte which is interacting for the first time, this is called as neo-lymphocytes. Now this naive lymphocyte interacts with the antigen, generates primary immune response, and along with the primary immune responses produce effector cells and memory cells. Now these memory cells will generate secondary immune response whenever the same antigen comes subsequently. Likewise, there are certain questions ki how do memory cells are different from effector cells? Memory cells, you know, they are developed after primary or during the primary immune response. Memory cells are more numerous. Memory cells are responsible for the primary immune response. It is not like that. Memory cell attacks invaders. Effector cells do not. It is not like that. Memory cells live longer. Yes, memory cells live longer. Suppose a child is vaccinated at the age of 6 months and it generates memory cells during the primary immune response, means whenever it is being vaccinated. Now if that particular child lives up to the age of 100, this memory cell would be there throughout his age, throughout its life. So memory cells live much longer, even for decades, 50 years, 80 years, 90 years, as the case may be. During this time, they do not enter into the cell cycle. Sometimes examiner may ask, ki memory cells are in which stage of their cell cycle? So memory cells are in the G0 phase of their cell cycle. This question can also be being asked. Now, there is another thing that is being asked about vaccination. The idea behind vaccination is to induce what? Without the vaccinated individual having to get sick. So actually what we are developing when we are vaccinating a particular individual? Option A, passive immunity. Option B, the primary immune response. Option C is anaphylactic shock. And option D, non-specific defense. Obviously, the idea behind vaccination is to induce the primary immune response, because of which memory cells are generated, and memory cells will provide us immunity throughout our life, as we have already discussed in the previous slides. Now, there comes a very important concept that is known as clonal selection theory. Niels Jerne, David Talmage, and McFarlane Burnett, they proposed this clonal selection theory. Now, we'll see the question first and then we'll be discussing the entire concept. The question says, according to clonal selection theory, an antibody changes its shape according to the antigen it meets. An individual contains only one type of B cells. There are many types of B cells, and the one which comes into action depends upon antigen for which it is specific. Option D, each B cell produces many kinds of antibodies. Now, option C is correct. Let me repeat it. There are many types of B cells, and the one which comes into action depends upon the antigen for which it is specific. That means whenever a particular antigen comes into our body, we are having vast variety of B cells in our body. Now, a particular B cell that is specific for that particular antigen would be selected. It will expand. It will go for clonal division. It will produce its clones. And then this entire big number of that particular B cell whose antigen has entered into our body, it will generate immune response against that. So, a vast array of cells are being there. From that, one or few cells are selected which are specific for particular antigen, and they will bring about the immune response. This is known as clonal selection theory. It is one of the very important concepts during in the process of development of immune response. Here comes B lymphocytes and T lymphocytes. B lymphocytes, T lymphocytes, they are the components of acquired immunity. So needless to say that they will generate immune response only when antigen will enter into our body. They have specificity, they have diversity, they can recognize between self and non-self, and immune response would be generated only against the non-self molecules. If we see a comparison, B lymphocytes, they develop and mature in bone marrow. T lymphocytes, they will develop and mature in thymus. B lymphocytes, after antigenic stimulation, that is interaction with the antigen, will develop into plasma cells and memory cells, memory B cells, whereas after antigenic stimulation, These T lymphocytes, actually there are two kinds of T lymphocytes, T helper cells and TC cells. So they will develop CTLs or memory cells, T helper cells or memory cells. These B lymphocytes, they produce antibodies, whereas T helper cells, a kind of T lymphocyte that is known as T helper cells, they will produce or secrete cytokines. B lymphocytes, they possess MSC class II molecules, and that is why they can serve as APCs. T lymphocytes do not have MSC class II molecules. Besides that, some other receptors are present, for example B7, BCR, CD40, etc. In the case of T lymphocytes, a specific TCR is present, that is known as T cell receptor, along with the molecule that is known as CD3 molecule, CD28 and CD45. Now, one important distinction between B lymphocytes and T lymphocytes. B lymphocytes can directly bind with the antigen. If an antigen comes, B lymphocytes can directly bind to it. But T lymphocytes cannot directly bind to the antigen. T lymphocytes can bind with the antigen only when the antigen is being first processed, either by any of the nucleated cells or by APCs. And then this antigen has to be presented with appropriate MHC molecule. We will be discussing what is MHC molecule, major histocompatibility complex. So when a specific MHC molecule will present the antigenic peptide, then only T helper cell will interact with it. So in that sense, T lymphocytes will form a ternary complex, that is TCR, antigenic peptide and MHC molecule, whereas B cell can directly bind to the antigen. So they will form a binary complex. So it is one of the important distinctions between B and T lymphocytes. T cells are, or T lymphocytes are MSC restricted, whereas B cells are not MSC restricted. Besides that, B lymphocytes, they will produce antibodies. T lymphocytes, there are two kinds of T lymphocytes. T helper cell will produce cytokines, whereas CTLs, that is cytotoxic T lymphocytes, they will kill altered self cells. So there are two different kind of lineages of T lymphocytes. These are the important differences in the B and the T lymphocytes that we have already discussed. This is the diagram of B and T lymphocytes, and you can see very well that B lymphocytes, they are having antibodies on their surfaces, and these antibodies are called as membrane bound immunoglobulins. In fact, IgM and IgD molecules can be present as a receptor on the surface of B cell. These are very specific receptors and they are known as BCRs. They will specifically interact with the antigenic epitope. Likewise, T lymphocytes, for example T helper cells, they have specific TCRs, and these TCRs will specifically interact with antigenic peptide along with MHC molecule. Here it is noteworthy that all the T helper cells are CD4 positive and they do not have CD8 on their surface. On the contrary, all the TC cells, they are having CD8 molecules on their surface and they do not have CD4 on their surface. So T helper cells are called as CD4 positive T helper cells, and TC cells are known as CD8 positive TC cells. Besides that, TCR is common. The T helper cells are MSC class II restricted, that is they will interact with the antigen only when the antigenic peptide is bought with MSC class II molecule, whereas TC cells will interact with the antigenic peptide when it is being bought with MSC class I molecule. This phenomena is called as MSC restriction. We will be discussing it in later slides. Let us first see some few of the questions. Which of the following statement is correct? T helper cell expresses surface CD8 receptors? No, because T helper cell have CD4. T cytotoxic cell express CD4 receptors? No, actually they have CD8. T helper cell expresses surface immunoglobulin molecules? No, surface immunoglobulin molecules are present on the B cells. T cytotoxic cell expresses surface CD8 receptors. Yes, this is the right option, that is D, because we know that only TC cells are having CD8 receptors on their surface. This particular concept we have to remember. Now, among the types of immunity, there are two kinds of immunity, based on whether cells are directly involved in the process of elimination of the antigen or cells are producing certain molecules. Here we have to see the root of humoral immunity. Humoral word is being derived from humor, that is body fluids. So if we are having immunity which is because of body fluids or the molecules which are present in the body fluids, then it is called as humoral immunity. And it will include components such as antibodies, complement, stuffs like that. It was first of all proposed by Behring, and it is more effective in the case of bacterial infections. On the other hand, cell-mediated immunity, here the immunity will be because of specific kinds of cells. For example, macrophages, NK cells, CTLs. So we see direct cells are involved. These cells go and kill the pathogen or eliminate the antigen. They are not producing certain molecules which are doing the job. Themselves they are doing the job. So when the cells are themselves involved in the process of providing the immunity or defense against the pathogen, it is called as cell-mediated immunity. And this entire concept of cell-mediated immunity first of all was being explained by Machnikov. And this particular kind of immunity is very much effective in viral infections and in the tumor immunity, that is against tumors. This is a diagram that is being taken from Janus Cubi, and this diagram explains that you can see T helper cell, they interact with MSC class 2 molecules, whereas TC cells, they will interact with antigenic peptide when it is presented with MSC class 1 molecule. T cells are MSC restricted. TC cells are restricted by MSC class 1 molecule. T helper cells are restricted by MSC class 2 molecules. On the other hand, if you see at the B lymphocyte level, B lymphocytes, they are having BCRs, that is B cell receptors, that can directly bind with the antigen. And after that, it will develop plasma cells. These plasma cells will be producing antibodies which will eliminate the antigen. And here you can see these antibodies are identical to the surface receptors. So this particular antibodies are called as secreted antibodies, and these antibodies which are present as a receptor on the surface of B cell, it is called as membrane bound immunoglobulin. So there are two kinds of immunoglobulin molecules, one which are present on the surface of B cell, you can see here, and those which are present as secretory molecules. These are secretory antibodies, these are membrane bound antibodies. Now we see certain examples. The biggest difference between cell mediated and humoral immunity is option A, how long their protection lasts. Option B, whether a subsequent secondary immune response can occur. Third, whether clonal selection occurs. And fourth, how they respond to and dispose of the invaders. So option D is right, how they respond to and dispose of the invaders. In the case of humoral immunity, it is the molecules which are present in the body fluids, whereas in the case of cell-mediated immunity, directly cells are involved. These cells will go and by variety of mechanism, they will directly eliminate or kill the pathogen. Now in the next part, we will be discussing the antibodies. Antibodies are also called as immunoglobulins. These are present on the surface of B cell membranes, and they are secreted by plasma cells. These membrane-bound antibodies will confer the antigenic specificity to B cells, and antigen-specific proliferation of B cells would be elicited by the interaction of membrane antibody with the antigen. Now here comes a very interesting thing, why antibodies are known as immunoglobulins. Antibodies are known as immunoglobulins because they belong to, these are the serum proteins which belongs to the gamma globulin fraction of the serum. And since the gamma globulin fraction of the serum is involved in immune functions, such molecules are called as immunoglobulins. So by this time we have discussed various kinds of concepts which are involved in the immunity. We have discussed various types of immunity. We have discussed antigens, epitopes, clonal selection theory. In the next section, we will be discussing the antibodies, their types, functions, and how they work on the antigen. Thank you.